Hello, class. This will be the second half of a lecture I gave in person, but I'm going to record it again here. So, second part of the cells chapter. Here we go. All right. So we ended up talking about the cell membrane. Plasma membrane, cell membrane, just so important to all the functioning of a cell. It's the gateway for what things get in, what things come out. Signaling molecules are going to come and tell the cell what to do on those proteins that are on the cell membrane. So it's made up of these phospholipids, right? Fatty tails, which will all be facing each other, and the hydrophilic heads, the phosphate part towards the water. And so even if you throw a bunch of phospholipids into water, they all arrange themselves like this. So they make these spheres, which certainly look like protocells. They don't have DNA, and they don't have other things that make them life, but they certainly have the look of a cell. And so you can see how early in evolution, how a phospholipid bilayer would be a beautiful cell membrane because it just forms itself naturally. So the important part on this plasma membrane is not really the phospholipids. They're all over the place. And that tells you that the wall is fatty. So fat-based hormones, steroid hormones can get right through, but charged molecules, big molecules, they can't get through that because the oil and water don't mix. So these proteins that are stuck in it, some of them are stuck on the inside, some on the outside, and some are transmembrane that go across. And the interesting thing about it too, if you look at transmembrane, is that the amino acids that make up this protein, you'll see that you have the charged ones on the outside and really the uncharged ones in the middle. And it kind of keeps it anchored in there because as soon as it goes too far up or too far down, those two ends there get kind of repelled from the inside of the phospholipid layer. So that's kind of cool. And these things move around. We know that. You can take some of these proteins, label them, and then put them on one side of a cell, come back a day later, and they're scattered throughout the cell. So they're like glaciers that float in the ocean. They can go all over the place. Looking at these proteins, we can kind of break them down into some important ones here. Receptor proteins are found on surfaces of cells, and they have a lock that is ready for a key, which will be called a ligand, or a hormone will bind to it, just fits into it, and it's going to have some kind of effect on the cell. Usually it turns on an enzyme, or it excites another messenger, then another messenger, and it makes some effect in the cell. So cells are waiting for that signal, and then they do something because they have proteins that have just the right shape, that they have that lock that is just waiting for the right key. Recognition proteins, all of my cells have proteins that, of course, are made from my genes, right? Proteins come from the recipe in our genes that are unique to me. And so if I were to get a kidney transplant from someone else, I would have different proteins on it, and I would recognize it as foreign, and I may attack it. Transport proteins, oh, so important to let things in and out. And some are just holes that aquaporins just water through. Others of them are gated, and some of them take energy to open. But this is a way things can get in and out of the cell, that they can't just go through the membrane. They need to have these channels. And remember, enzymes are proteins. And so that's what we're talking about. Some on the inside, facing the intracellular fluid, some on the outside, and some are through. But sometimes they're turned on, and then they start doing their enzymatic processes. Otherwise, they're turned off. So I like this illustration down here. It shows how I picture that plasma membrane. Remember that plants will have a cell wall solid, but this membrane is like pushing on a waterbed. It gives and it comes back, so it's fluid. Actually, the phospholipids are rotating and moving, and then the proteins, some are kind of anchored, but other ones are just floating in the sea of phospholipids. And you can see all the extensions coming off. You get some carbs coming off some of the proteins that identify the cells and then do this and that. Very cool. I want to mention cell adhesion molecules real quickly. It's going to become important when we talk about the immune system. Let's say you get a cut and it gets infected and bacteria get into your skin. Well, the capillaries nearby. You guys remember what mast cells will secrete two chemicals that start with an H? Heparin and histamine that are going to make the blood flow faster and make the capillaries expand and become more leaky. So, and then also the cells will give off these little sticky things, or like that spike strip that police put that will stop the car because the tire will stick in it. So they put out these little sticky molecules, and then a white blood cell, a neutrophil is coming along, it's going to stick to it. It's like, okay, this is where I need to go, and then it will leave the bloodstream there. And these cell adhesion molecules are important early on, when embryonically, when we develop our bodies to kind of guide where you want certain processes to go. All right, as another pathology, it was known even back in the 1600s that if a child, the skin tasted salty, the child wasn't going to survive. Of course, they didn't know what was going on. They didn't know what DNA was back then. But now we know. That means that you have a mutation, a genetic disease, where you don't make one of those proteins that's going to pump chloride, a little bit sodium. So it's a salt issue. So what's the big deal? Well, it makes your mucus really thick. And so it clogs up your pancreatic ducts, so digestion problems, but especially respiratory problems. Your lungs fill with this thick mucus because the protein is just not the right shape, and it doesn't function properly. Yeah. And they used to take babies and kids and they just beat their chest every morning to loosen up that mucus. Now they even have vests that kind of can physically do that too. And then one more thing, looking at an example of proteins, is looking at beta blockers. So these are drugs, and they mimic, in this case, adrenaline or epinephrine. It means the same thing. And this mimic is going to, well, normally when you get anxious or get a fight or flight response, adrenaline will be released, and then it'll bind to, for instance, these proteins that are in the pacemaker of your heart, and they'll speed up your heart, right? Well, like propranolol, yeah, the drug is a beta blocker, will go in and it will gum up all those receptors. So even though you're awash with adrenaline, you don't respond and your heart levels stay low. So it helps keep your blood pressure down, or with anxiety, it'll keep your blood down, even if you've got this adrenaline. That's cool. So a lot of drugs, they work on these proteins in the synapse between neurons. You've got depressants like alcohol, they're going to go in, or things that are excitatory, like cocaine and amphetamines, things like that, that can work on these receptors by either mimicking the hormone or blocking it, or screwing up the recycling of it. And so it's how a lot of drugs work. You need to know these proteins and the shape of it and their effect. All right, now if you imagine a cell like fills the room that you're in, or the lecture hall, you wouldn't really be able to see all the way across that lecture hall in real life, because in the cell, you have this scaffolding of tubules and girders everywhere, that cytoskeleton. And it provides structure, because remember, animal cells don't have a cell wall, it's just this fluid membrane, provides some structure. And also a lot of transport in the cell. You'll see substances will move along these tubes to go to different places. But you've got this skeleton that you don't normally see, but you can stain for it, especially here at stain. You see, oh my God, look at that. There's tubes everywhere. It's filling up the cell. So the scaffolding is the cytoskeleton that is important, but you don't see it normally. All right, you all know the nucleus, the brain of the cell. It's where you keep your DNA. And your DNA has all the genes, 25,000 genes that make all the recipes. It turns out there's like 250,000 of those, because you can one recipe can kind of be tweaked and you can get some different products. But so it makes all the proteins, is what makes you human, is what's your cells are making. The recipe comes in your genes that makes you. And you don't want that master recipe to be all scuffed up out in the cytoplasm. It's used and used and used. You keep it in the vault, in that nucleus, and you just make copies of it, and you bring it out to the cytoplasm. There you can make the proteins, but let's keep that recipe, tight in the nucleus. So that's it. The nucleus holds your DNA, and it's not just naked in there, especially in us eukaryotes. It's wrapped around the proteins, these histones. You've got to compact it. Because it's 3.5 billion letters long. So we need to compact it. So we wrap it around histones, and we wrap it along. There's all these levels of compaction, so we could fit all this long DNA into our nucleus. And also you have most of the genes, remember, are in storage. You know, think about different cells in your body. They're not all the genes, all of them are being expressed. Only a certain number of them are. When you want to express a gene, you've got to make it available. You got to make it naked. So you got to unpack it. And so if this was a DNA of a pancreatic cell that's making insulin, the recipe would be held open. And then, you know, over here, the recipe to make keratin, to make your, you know, your skin or your hair, that's in storage. It's in a box. It's unavailable there. Yeah. And the nucleus, yeah, again, it's something you can't miss. I mean, like if it was in our lecture hall, I mentioned it'd be like the size of a full-size garbage truck or dump truck. It's huge, right? The nucleus. And you can see here in this lymphocyte, that dark purple makes up most of the cell. That's the nucleus. And there's an electron micrograph here. And you notice these dots. Yeah, that's the nucleolus. And you won't see it in all nuclei, but you find it in those cells that are making a lot of proteins, because the nucleolus is where you make ribosomes. Ribosomes, I'll talk about soon, are where you make proteins. So if you're looking at a liver cell, even our kidney cells we looked at, they're very actively making enzymes or hormones or proteins of any kind. We'll have a big nucleolus. And then in your cartilage cell in your ear, you don't see nucleolus because the cell's not doing much, it's not making much. And you can see the nuclear membrane has pores in it. That's how things get in and out. All right, so in a lab, we looked at pseudostratified columnar that had cilia, these little hairs that would move. The mucus would trap the dust and bacteria, and the cilia would move it up where you'd cough it or swallow it, yeah. And just as an aside, in our columnar cells in our gut, we have little extensions, but they're called microvilli. And these don't move. They're just little extensions, little fingers that increase surface area. So put that aside. These flagella and cilia, they beat, they move. There's tubes in there that slide, and it causes it to move. Now, there's no difference between flagella and cilia structurally. It's just that when you only have 1 or 2 or 3, we call it a flagella, like a tail. When you have hundreds, we call it cilia. That's all. If you look inside, they all have this 2 in the middle, then you have 9 pairs on the outside. I won't draw them all. And what's so cool about this is that that's our cilia, like in my trachea. It's also, if you looked at a flagella of sperm, or you look at a bacteria or a protozoan, even some plants, the gametes swim with a flagella, same 9+2 structure. And what's cool about it is that clearly we all evolved from an ancestor that had this flagella. We all inherited it, because you wouldn't have us and bacteria and plants independently come up with the same 9+2 structure. That's just one way of making it. It would never have come up by chance in all these groups. So it's more evidence of evolution from a common ancestor. All right. One thing you think about if you had a cilia issue is what would be what would be the symptom? Imagine you had genetic disease where you don't make the cilia correctly, so they don't move. I'll wait. You're not in front of me, I'm talking to a computer, so I would not wait anymore. You would have respiratory problems. Just like when you smoke, it kills the cilia, you have to cough all the time to get that mucus up, same thing. And I mentioned a woman just to also make you think about your body too. What would be another symptom of a woman besides having respiratory problems? She'd also be infertile, because in the fallopian tubes or oviducts, the cilia move the egg down. And so they don't in people with cilia issues. All right. Mitochondria, one of my favorites, bizarre, interesting organelles. What do you guys, when you think mitochondria, what do you think about? I mean, first thing comes to my mind is that whole cool thing how, I'm going to just tell you, they used to be free-living bacteria, right? That are now living symbiotically within our cells. Yeah, they got their own DNA. In terms of anatomy, it's important because we'll talk about it the next chapter, is that I think of it like taking a plastic bag from the grocery store, crumpling it up and putting it in another bag, plastic bag. And so you have this outer membrane and then this inner membrane that's really folded and crazy, right? And on that inner membrane is where we're going to see where we do this electron transport chain, this where we convert sugars finally into ATP. So the purpose of our mitochondria for our cells is that they take sugar, they're fed sugar and oxygen, and they give us ATP, which is the energy source we need to run our reactions, to make our muscles contract. So yeah, they're little powerhouses. And those cells that use a lot of ATP, like muscle cells, have thousands of mitochondria. And those that are quiescent and not being used have fewer. And you can even with training, aerobic training, you can do a muscle biopsy, count the mitochondria, and then have maybe take two groups, have one do aerobic exercise, long distance running, biking, something like that. The other group just does maybe weightlifting and sprints. So yeah, and what you'll see if you take that biopsy like 6 months later is that those that did aerobic training, their mitochondria increased because they do that. If you do anaerobic training, you don't need to make ATP. You don't need oxygen if it's really kind of rapidly. We'll get into that with muscles. And of course, once again, we know these bacteria that were free living were engulfed by some early cell, and instead of eating them, they hung out like we didn't eat them. And all of a sudden we realized, hey, I'll give you some sugar, a place to live. You just give me some ATP. And this relationship happened. Plants have mitochondria, so it happened before plants and animals split that long ago, that these things have been living with us inside of us. They're the same size as bacteria. They have a lot of characteristics. Their DNA is circular like a bacteria. They've lost a lot of genes. They have few genes, one's mostly related to aerobic respiration because they've lived symbiotically with us for so long. They don't need to know how to live in the outside world kind of thing. Yeah. And so, when you're asked this question, really, well, first of all, what would a mitochondrial disease, what would be some symptoms? There are mitochondrial diseases. Yeah, fatigue. You don't make enough energy, so definitely. And can you blame dad for that genetic disease? Dad's off the hook. It's mom's fault, grandma's fault, because you start out with an egg, and that has the mitochondria in it. Dad's sperm just gives the DNA. It doesn't get mitochondria. And I say that with one caveat, that there's been a recent study last few years that we see some of the male mitochondrial DNA found in the offspring. And so there's some, we're not sure how that's happening, but normally mitochondrial DNA is maternally inherited. It comes down the female because it's found in the egg, not in the sperm. And we use mitochondrial DNA and nuclear DNA. Both of those can be used. They evolve at different rates, and it depends on the question you're asking. But mitochondrial DNA has been used to look for mitochondrial Eve, in Africa where people originated. You can follow the changes as people migrated. All right, that's it for mitochondria. Now, lysosomes are the vesicles or sacs filled with these enzymes that break down everything. So if your body, if you want to consume something or get rid of some garbage, you take that, say that bacteria you've taken in, you take a lysosome and you fuse them together, and the lysosome dumps all these enzymes in around the bacteria, and it's going to break it down so you can recycle those nutrients. So lysosomes are filled with some really toxic stuff that can, they can just break down everything. So it's kind of dangerous. It's like having a big beaker filled with acid in your dorm room. It's like, if you knocked it over, it would start dissolving your desk, right? So there are some diseases where the lysosomes burst and they eat away the cells themselves. So we keep these things, the chemicals in these things, and we just use them carefully. Yeah. Another disease is Tay-Sachs disease, genetic disease, very, very sad, because what happens is you have a child about 6 months old starts showing neurological problems. And then what you see is that child won't survive past 4 years old, as a child. So, and there's nothing you can do. And this is the issue. The issue is there's a genetic problem with one of these enzymes in the lysosome that's supposed to break down this kind of fat. And without it, the fat builds up. It's like if no one takes out the garbage, it just keeps building up in the kitchen, right? But this is a fat in your brain and spinal cord. It fills up the cells, and In the beginning, it just kind of builds up, but then you start seeing symptoms, and then, yeah. So it's pretty rare now. It's found in like Eastern European Jewish community, and I think Canadian, French Canadians. There's some strange groups where it's like disparate groups where it's the gene is most prevalent, and if you're in that kind of group, they could do genetic testing and see if you carry that disorder. All right, so let's look at some other things. The ER, not the emergency room. This is the endoplasmic reticulum. You have a whole bunch of, if you look in a cell, a whole bunch of membrane areas that are going to be very important. So I'm looking at rough ER here. See those little purple circles? This is an electron micrograph, and then they colorized it. The colors aren't real. But those little purple circles are the ribosomes. So I know that's where I make proteins, and they're stuck all over this yellow membrane system. So I'm going to make the proteins in the ribosomes. They go in that yellow membrane system where I can modify them, change them, put them together, fold them, do whatever I'm going to do with them. Where's a ribosome? So it's actually made of two parts. It comes together, and ribosomes, they float around your cytoplasm, but they're also stuck on the ER that makes it rough ER, because it's rough because it has little dots on it. And what happens in a ribosome is that the messenger RNA goes through it, and it's read in groups of 3, and the right amino acid is put out of it. So the message goes through this way, and the protein gets spit out the top. So it's where we translate the genetic code into proteins. And rough ER is a membrane system that's all studded with ribosomes. So you're making proteins right there. And it's right next to the nucleus too. So the messenger RNA has just come out, and then it can be read by ribosomes. And they can be read numerous times, the same message, and you're just pumping out that product. And then the rough ER will be able to modify it. Yeah, so we know that's, we can kind of follow this, how the recipe is in the genes. It goes out. We know it goes to the rough ER. It's going to leave there. It usually goes to the Golgi apparatus I'll talk about next. And that's going to be modify the product more and ship it out to wherever it's going. A smooth ER has no ribosomes, so it doesn't make any proteins, but it's going to make fats. And I also need you to remember that the smooth ER is real important in detoxifying toxins like alcohol or illicit drugs and normal drugs. Anything that's stored in your body, that smooth ER will break it down. Yeah, and actually you can work out your liver just like you work out at the gym. Just keep drinking, alcoholics drink, and the liver inside of it, the cells get more and more smooth ER. And so a person that is really drinks a lot, it takes more alcohol to get them intoxicated because they've got so much smooth ER that tries to break down the alcohol. Eventually you can overwhelm it, of course, but yeah. What I also want to mention here is that if you're that kind of person that has really assaulted your liver so that it has a lot of smooth ER, other drugs, even like penicillin, antibiotics, things like that, they're also broken down too rapidly. So yeah, by having a lot of smooth ER, your liver is ready for toxins, which in some cases can be any foreign substance, including medicines that you want to get rid of. So it's something you keep in mind when you're dosing someone. Yeah, there you go. All right, Golgi, it's an Italian scientist that it's the mailroom. And so that means it has a surface that's like towards the nucleus in the ER and a surface that's towards the outside of the cell. And things come in one side and they go through this where they're further modified, packaged, and they're given their destination. Are they going to go to the mitochondria? Are they going to go outside the cell? That's the deal. Yeah, so that's the kind of show you what's going on. Smooth ER making some lipids, rough ER making proteins, Golgi putting them together sometimes or modifying them and sending them on their way. Peroxisomes, just talk real quickly. They're also found in your cells, and there are some diseases that people have problems with these. But they break down substances in your cells, some toxic things. And I want you to remember that it often makes hydrogen peroxide, which is nasty. The oxygen can oxidize things and get in the way of reactions, right? And hydrogen peroxide is just water with an extra oxygen. We keep it in dark bottles because sunlight breaks it down to water and oxygen. And I want you to understand why we use hydrogen peroxide. Unlike Neosporin or alcohol that, are going to kill the bacteria. This doesn't do that. In all of your tissues, you have an enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. And so when you have a cut, you put hydrogen peroxide and it foams up, doesn't it? And that's the sole purpose. What you are doing is you are making the oxygen is going to dislodge any kind of dirt and such and bring it to the surface. So hydrogen peroxide foams it up because of the catalase that we find, and it cleans out the wound in that way. All right, so a few more things in this chapter. Make sure we understand for A&P, we get the basics here. There are cellular processes that don't require energy, things like diffusion, right, osmosis. And anything that is an active process requires energy, ATP. So diffusion always goes from a high to low concentration, right? You know that. Facilitated means it's got some kind of channel or helper to help it, but it's still no energy. Y'all know osmosis, I'll talk about that. And, filtration, I think of filter paper in chemistry, and it keeps the precipitate up here. But in our bodies, we have our blood pressure, and only certain things get through the capillaries, is what we're mostly talking about. And then active transport takes energy. Endocytosis, bring things in the cell. Exocytosis, exiting the cell. Transcytosis, bringing things across the cell. Basic vocabulary. Diffusion. I don't think I think you guys should understand that from biology 100 times over, right? High concentration, it's going to spread out and diffuse. Because, I don't know what that is. Because of all the molecules bouncing around, that Brownian movement that's going to cause diffusion, right? And this is how we get gases in our lungs. Oxygen, carbon dioxide, simple diffusion. It's so smart. It takes no energy. We just need a big wet membrane, and our lungs is the size of half a tennis court. And then have blood right underneath it. And then oxygen will diffuse into the blood and carbon dioxide out because it goes along the concentration gradient from high to low. A beautiful thing. No energy, and we're exchanging substances. Facilitated, like I mentioned, some things need channels to get through that membrane. And so it's still diffusion high to low, but it has some help. Osmosis, again, I put a little video on here, but you guys should be reviewing that, right? It's when water goes from high to low. I'm just going to leave it there. And of course, looking at our bodies, you wonder why we just hammer osmosis like you learn all the time. It's actually really important in A&P and in your body. When you're thirsty, what happens is that you have cells in your brain, hypothalamus, that when you're thirsty, when you're dehydrated, there's so much salts, your fluid is concentrated, that it sucks the water out of these cells in your brain because of osmosis. And the cell shrinks up like a raisin. And then those cells secretes these hormones like antidiuretic hormone, which makes you retain your pee, make you thirsty. On the other hand, if you drink so much water, you're overhydrated, those cells swell up and they're going to inhibit that and make you pee more and get rid of water. So you can see right there. And then when we look at when you get a IV bag kind of thing, when we look at blood cells, isotonic, it's at the same tonicity, yeah, whatever that is. It's the same concentration in and out, so water doesn't move either way. But you put it in a hypertonic, a salty solution, water will leave that membrane. And hypotonic, water will enter that blood cell and lyse it or burst it. So just some terms, make sure we're on the same page. All right, filtration needs a semi-permeable membrane and it needs some kind of force. In this case, it's blood pressure. So you see filtration in your kidneys and your capillaries, water, glucose, oxygen, they go right through. But blood cells don't, big proteins don't, because they can't get through those holes. And active transport takes energy. And when we use that, what we're doing is we're not going from high to low concentration. It's when you want to concentrate something. So you go from low to high. So you're going to be doing that. And so if you're going to do that, it's not happening naturally. It's not naturally going to happen. So you have to use energy to make that happen. So you'll see in our cells, we have this sodium-potassium pump. You guys can watch this on if you like, but it's got where we use ATP energy, we pump sodium out and potassium in. It goes both ways and it takes tons of energy, but we do that so we make this gradient of sodium and potassium. It's just an example, important example of some active transport. Again, endocytosis, bringing things into the cell. Cyto means cell. Pinocytosis is cell drinking just little bits. I think of a wine like a Pinot Noir or Pinot Grigio wines. It's just sipping a little bit of wine. The cells are sipping the environment. Phagocytosis is taking it a big chunk. And a special kind is called receptor-mediated endocytosis. And that is where if your cell wants to take in something specific like this, it will bind on the receptors, and that will cause the cell to take it in like that. And exocytosis is the opposite. It's a substance in the cell you want to get rid of. So it goes in a vesicle, binds with the membrane, and releases it. All right, well, I'm not going to go through the phases of mitosis. You've learned that. You better know that by now. I may ask you that, but I'm not going to spend time going over that. But some things I want to make sure you understand is that cells go through this cell cycle of dividing and not dividing, resting, being active, right? And it just depends. Your epithelial cells we talked about in your skin, lining your gut, they are constantly dividing. You know, you're constantly making those. And other cells like your neurons in your brain, your cartilage cells, they are not dividing. They're very slow. So cells get signals to whether they should divide or not, and they go through this cycle. What I want you to know is that during this interphase, this period is when the DNA is actually duplicated or synthesized. And by the time it enters prophase, the DNA has already been duplicated. And now at that point, you're just going to divvy up the material between the two daughter cells. But during that interphase is when the DNA is replicated. Okay. The other thing I want to point out here is that you have these checkpoints. And what happens here is that the cell checks its DNA. And if there is any damage, the cell kills itself. We'll see. Apoptosis is cell suicide, cell death. And so what's important in cancer is a couple checkpoints here. Cancer is that we screw up these checkpoints. This is one way you can do it. So that you don't check it, or you check it and you just don't care. So here you have this cell that's completely screwed up in its DNA, and it still divides. In a healthy person, any issue that can't be fixed, that cell, get rid of it. It's going to cause cancer, all right? So these checkpoints are important in the cell cycle. And it's one point where cancer can happen. So interphase is the time when the cell is not going through active division, which is mitosis. Yep. Again, prophase, metaphase, anaphase, telophase. Just know what's going on. Look at it over like, okay, I got this. Animation if you need it. All right, so cell division. Like I say, the cells in your body vary so much in how much they divide, right? Some are divided constantly and some are not. One thing that's interesting, we talk about aging. I don't know, we don't have time in this class to talk about everything, but at the ends of your chromosomes, you have these tips that are just a bunch of repeated nonsense. But every time your cell divides, they get shorter. A piece breaks off. They're called telomeres. And so it's believed that's like a clock that allows your cells to divide, like 50 times. And that's why we age, because once you end up with those telomeres are completely gnawed away, then you start breaking up good DNA. So these telomeres are fascinating, these sequences at the end that get shorter every time it divides. It's kind of like a clock as you get older. Very cool. And again, cells need to divide because you can't have a huge cell. It doesn't have enough surface area, so they need to keep small. Surface area to volume. We'll talk about that. And then how do they know to divide? Well, they're stimulated by factors that tell them to divide. Like red blood cells, they have this hormone EPO, which tells your bone marrow, make more red blood cells if you move to Denver or you bleed a lot. And normally cells, let's say I got a cut in my arm, the cells will fill up. And once they fill up the divot, they stop. Called this contact inhibition or density dependent, where when there's enough of them, they stop. But in cancer, they keep going and they'll make this tumor. So cancer and tumors are when there screws up the cell cycle and you just divide out of control. It's a disease unlike a lot of like infectious diseases in that it's not a foreign invader, it's your own cells that have gotten mistakes that have built up, and then they lose control of being able to kill off those mistakes, and the cells are dividing constantly when they shouldn't. Yeah. And if you hear benign, that's good. That means this tumor is localized. If it's malignant, it means it's growing. Let's put these fingers out. Cancer, like crab, the zodiac sign, they looked at early cancer, looks like the legs of a crab are like invading adjacent tissues. Yeah. And if it metastasizes, it means it's moving. So a person, you may say, oh, they got lung cancer, just cut out the cancer. That can work, but up to a point, because if it's already spread everywhere, you can't get rid of it everywhere. Yeah. And look at what causes it. Oncogenes cause cancer. Oncologist is cancer. On the other hand, tumor suppressors are the other side of it, where normally those checkpoints will stop it. So tumor suppressor genes are like taking the brake pedal and throwing it out of your car. You just keep going. All right, and then just finishing up, stem cells, you have them in your bone marrow. You started out as a fertilized egg. That fertilized egg is totipotent, totally potential. It can become anything. But as cells divide, they narrow their options, though it can become certain things. So pluripotent means plural. It can become many things. So your bone marrow stem cells can become any kind of blood cell, but they can't become a bone cell or a skin cell. Yeah. So you can see early on, it's totipotent. Stem cells become anything. And stem cells are so cool in research because, instead of Parkinson's, we can implant stem cells and make them grow normal neurons, we're hoping, or stem cells grow a new heart or a new kidney for you. So a lot of potential, because stem cells can become anything. But as an adult, our cells are committed. We don't have a lot of them. Yeah, we can make little mini brains. It can't think, but just letting you know, stem cells can grow whole organs. Yeah. The term apoptosis, you should know, it's programmed cell death. It's nothing to be scary about. It happens all the time. Our hand starts out as like a catcher's mitt, and then apoptosis kills the cells in between. You get a bad sunburn, and you kill those cells. It's your body notices the cells are screwed up or they're not necessary, and they're killed off. All right, class. Let's see. Cells. There should be a lot of review, and hopefully this lecture has helped. Thank you.